Hello, I'm Deb Sims. I'm a CLL patient from Melbourne in Australia here at ASH 2019 in Orlando for Lymphoma Australia and the Lymphoma Coalition. And I'm joined by Professor Miles Prince from the Epworth Hospital in Melbourne and the Peter Mac. Professor Prince, thank you for joining us. It's been a fantastic ASH. What have been some of the highlights for you?
It's been a brilliant ASH. It's been a very consolidating ASH. It's been not about new drugs, but it's been about drug combinations, and particularly with relation to lymphoma, drug combinations with CLL, follicular lymphoma. That's really been the highlights, and a lot around making the right diagnosis.
And that was what stood out for me as a patient. It seems to be that there's a more sensitive diagnostic testing going on, and that testing is helping guide clinicians with their treatment choices. We don't necessarily have all these tests. Is that something we're going to be taking back to Australia and fighting for, basically?
Ultimately, treating the patient needs to be based on having the right diagnosis. And now for conditions like CLL, diffuse large B-cell lymphoma, follicular lymphoma, they are different diseases within one larger group. And we know, for example, in CLL that broadly there's a group of patients that fit into what are called mutated and unmutated, and then there's patients, the rare group, that has P53 mutated. So these mutational tests, looking for what I would call the abnormal note in the song of CLL, that one single abnormalities that can occur can make a huge difference in terms of the patient's response to treatment and choice of treatment. So I think it's absolutely necessary, and I have a strong belief that anybody with CLL who requires therapy needs to have molecular testing, this mutational testing, as an absolute must to guide their treatment both in the short term and into the longer term.
Yeah, and one thing that I stood out to me was that if you have a certain type of CLL, the unmutated and 17p deleted, you should not be having chemotherapy. And this is something that we are still giving in Australia, regardless of that.
Well, I think that the other side of the coin when we're talking about treatment is the toxicities. So people always talk about efficacy and responses. What we know with P53 mutated CLL is that often patients will respond, but they go through months of treatment, chemotherapy, only to relapse quickly. So they go through all the toxicities. So why would you do that when you know that it's going to be a short response? And so that raises the question, well, what's the alternative? Well, the alternative is the new drugs, Ibrutinib and venetoclax. And into the future, we're going to be talking about combinations. But ultimately, these drugs work better than chemotherapy in that subset of patients, and therefore the doctors need to know. At the moment, Ibrutinib isn't available up front in places like Australia, but knowing that the chemotherapy is going to have only a short response may influence how much chemotherapy you give, how closely you monitor the patient, and how quickly you might switch to a drug like Ibrutinib.
I'm very passionate about clinical trials. I've benefited from a phase 1 clinical trial of venetoclax and obinutuzumab. I was very lucky, got 4 years out of that treatment. We're hearing new molecules in all these diseases. Realistically, the only way we're going to get them in the short term as patients in Australia is through clinical trials. It's part of the role of our doctors here over the weekend is really to find out about what trials they can bring to Australia.
Well, I think that the only way we can move forward is by patients going on to clinical trials if they're eligible. People have got to learn not to be afraid of clinical trials, and they're not guinea pigs. They are not being tested with and missing out on the proper treatment. Ultimately, clinical trials are designed to make steps forward without disadvantaging the patient. And so we are very lucky in Australia compared to many other countries around the world where drug companies and larger investigational groups from the US or Europe or the UK want to collaborate with us and answer the big questions. Because fortunately, we've come a long way, and some of the questions are actually quite small and take a lot of patients to go onto the trials to do that. And we want to make these advances without having to be guessing for the next 10 or 15 years. So yeah, absolutely, it's important for us to be able to get clinical trials and then spread the word.
Finally, the cure for all of us with lymphoma, and particularly the indolent incurable lymphomas, is something that could be on the horizon with CAR T. And you've obviously had some experience with CAR T. What have you heard over the past weekend that has made you feel this is more promising?
Well, I think if we think about CAR T cells as in a parallel to how we've learned to fly in aircraft, we have come past the Wright brothers. We're somewhere between the First and Second World Wars. We've got these things up in the air, and at the moment there's some that have significant side effects, others where we're seeing not much responses. We've got different patient populations. So first of all, they work. They are a dramatic paradigm shift. It's now about working out which patients are going to benefit from them. But if I was to say, what do we know? We know that they definitely work in acute lymphoblastic leukemia. They definitely work in large cell lymphoma. They definitely work in myeloma, and they definitely work in CLL and follicular lymphoma. They're going to be working. In the latter two, it's a matter of where do they fit. While there's a whole lot of changes in the other treatment. We don't want to use them too late, but equally they have toxicities, and we don't want to be putting patients at risk. Cure is the ultimate gain, but we have to make that an acceptable risk to the patient. And at the moment, CAR T cells, I think, there will be a lot of changes and a lot of improvements in the next couple of years.
Professor Prince, thank you very much for joining us. Enjoy the rest of ASH.
Thanks, Deb. Thanks very much.